Okay, so uh, today is the second week of the live session. So, welcome everyone. My name is Shuma. I'm the PMRFTA for this course, NGS Data Analysis and Applications. So, last week we talked about mostly different types of sequences, how they perform, what kind of outputs they give, and how we can interpret those outputs. This week we'll talk a little bit about after we get the output, what format do we get the output in the different data formats, and how we handle different data formats, and how we encode different data formats, and ultimately how like these can lead to the further downstream analysis. Okay, so let's get started. So this slide shows you a general overview of NGS data analysis workflow. So at the top left, you have the signal analysis part. So basically your sequencer, after sequencing the DNA, whatever you've given is and given it as input, gives you some sort of signal. So for example, it can be a fluorescent light signal, it can be a change in current signal as we saw from nanopore sequencing as well. But the signal can be varied across different platforms as we saw last time. But the whole idea is the whole sequencing is based on uh, like the output will be in some form of signal, be it fluorescence, be it radioactivity, be it difference in uh, ion flow or anything. So it is given in form of a signal, and there is a base calling software which basically identifies this signal and uh, uh, allocates a base to the particular signal. So if a signal is in a particular format, it will predict it to be a or a T or a G or a C depending on the different signatures of these particular bases, and how uh, how each of these individual bases give out the signature signals. So you have the base calling software here. After that, we'll go through these today mainly. So after base calling, you have base quality scores. According to the base calls, you generate certain uh, quality scores of the base, which tells you what is the probability that call is right or wrong. After that, you generate what we call as a FASTQ file. We'll look into what a FASTQ file is. But yeah, uh, after uh, generation of the FASTQ file, you filter. Basically, you do basic quality control. You check the quality of the reads, how it is. Then you filter those that are erroneous or that are contamination or whatever. That is actually not good data. Basically you filter those out, and then you do some sort of trimming of the data in cases of Illumina, if you remember. So we have two adapter sequences on both sides of the lead, which basically goes and binds to the probe that is present in the lane. So you need to trim out those adapter sequences. Yeah. So depending on your data set, you need to trim out your adapter sequences, and after that, what you get is basically a clean FASTQ file. So on top of that, like this part of the yellow part of the workflow is basically what is alignment and variant calling. So after you get a clean readout of your sequence, what you do is try to align it with a reference one. So the reference can vary. So we have different builds of reference genome. We have GSS 37, GSS 38, and very recently a new T2T consortium has generated a reference which actually resolved the whole Y chromosome. The paper that I shared last time. So you have different kinds of these references. You try to align with them, and post alignment you use a software that can identify the gaps where the alignment is not proper. Is that a variant? Is it a single nucleotide variant, or is it a big variant? Is it a structural variant above 50 base pairs or something? So according to the alignment, after the alignment is done, you use this variant calling softwares. There are different kinds of these softwares. We'll talk about it when we reach there in the next weeks. But yeah, so So first you have the signal analysis, then you generate your FASTQ, do some certain QC on your reads, then you generate the clean FASTQ file on which you do either you do read alignment, and if your reference is not present, say for example you have gotten a sequence, gotten the DNA from an organism which does not have a standard reference available to you. So what you need to do is what we call as de novo assembly. So from the reads only, without the help of a reference genome, we try to make up the whole genome from end to end. It's a very computationally intensive and a very difficult thing to do. Basically, a lot of algorithms, a lot of uh, graph theory algorithms, particularly, are used to do that, but it's erroneous and it's not, and it's very computationally intensive workflow. Anyway, so after that you do variant calling, and after uh, variant calling, what you generate is called as a VCF file, on which you annotate these variants based on their impact on the whole system. So basically, a variant might be a particular type of mutation which has been previously seen to be associated with a particular disease. So if you identify that variant in your sample, you may annotate it with a particular associated disease or a particular associated phenotype, to be clear. And after annotation, you do some post annotation filtering and you interpret your results, and that's the whole workflow for getting till the variance and the impact of the variance. Now, in this annotation part, what you can do is you can just a second. So different kinds of annotation you can do. As I said, you can do gene-based annotation. So a particular gene might have multiple variants, and depending on that, you may also depending on on the number of variants on a gene, a particular disease might be the person might be prone to a particular disease or anything else of that sort. So you can do all this kind of analysis after you have gotten your variant calling file, which is basically the VCF file, and on top of that you can do this like extra analysis, downstream analysis, which includes like how different genes will be regulated because of the variant, how protein function will be modified because of the gradient, various things you can do. So this was just the whole overview of the whole basically NGS analysis process. We'll go into details like at each part of this process. So today we'll be basically focusing on the left part, which is from signal generation to base calling to different kind of filtering that we do to get the best data uh, out of whatever raw data you have gotten. So today we'll be focusing on the left part. In the coming upcoming weeks, we'll go through these parts as well. So don't worry about that. Okay. So as I was saying, so after you have generated your basically read file, read file that you've generated. Now the question is, do you do an alignment or do you do a do you do a direct mapping alignment, or do you try to assemble without a reference? So that depends on the availability of your reference. So when your reference is available, it is always better to go with basically read alignment algorithm, because uh, those will be less intensive in terms of computational power and also time and resources. And if you don't have the if you do not have an idea about what kind of reference you need, basically the organism you're trying to sequence does not have a standardized reference genome. So in that case, you have no other way to but to assemble it in a de novo fashion from scratch. So once high quality data is obtained from pre-processing, the next step is read mapping or alignment. There are two main options depending on the availability of the genome sequence. When studying an organism with a reference genome, it is possible to infer which transcripts expressed by mapping the reads to the reference genome, which is known as genome mapping. If you do the same thing with transcriptome reads, it's called as transcriptome mapping. So in transcriptomics, what they do is they generate these particular transcripts, which is basically mRNA. And what they do is they convert it, they use retro viral mechanisms to convert the mRNA to DNA, and then they do the read mapping. So it's not like we do not have a kind of an mRNA to mRNA mapping. So if you are trying to study mRNA, what you need to do is convert it back to DNA using retroviral factors and enzymes, and you can align it to the your genomic reference. So that is how it is done. And mapping reads to the genome requires no knowledge of the set of transcribed regions or the way in which the exons are spliced together. This approach allows for discovery of new and unannotated transcripts. So we'll come to a little bit of detail in the next few classes when we're studying different types of transcriptomic techniques. But uh, basically this is how it is done. So if you recall, mRNA, the protein coding part of mRNA is the exons. So basically you have your gene, the whole gene is transcribed into an mRNA. After the transcription, what happens is your introns are cut off and they are taken out, and the exons are fused together to form the whole protein coding mRNA. Now if you just have the protein coding mRNA, it is very hard to find, because like it is continuous in the mRNA form, but in the genome, in the genome, or in your basically genomic data, the sequence of this mRNA will not be in a continuous fashion, because only the exons are present in the mRNA. So your exons will go and match, but in between the exons there will be certain gaps, which will be basically the introns, and there can be other factors in it as well. So uh, it becomes difficult to do a de novo assembly in that case. So we rely more on read alignment. And if you are working on an organism without a reference genome, you have no other option, but the reads needs to be assembled first into the longer context, which is the process which is called as de novo assembly, and this context can then be considered as the express transcriptome to which the reads are remapped for quantification. So basically, you'll try to make a big, longer stretch of nucleotides, or basically a longer stretch of nucleotides, and try each read and add each read to another, and try to find what is the best possible basically longest sequence that you can get. So it's not that always the reads will be like end to end, it can overlap. So there is another question of like basically computational algorithms when you're trying to use them. Like, are they considering overlaps, are they not? So these kind of things you need to think about it then. Uh, we'll talk a lot more about mapping in the next class, because in that we'll cover mapping. I just wanted to give you an idea about what we do, how we do mapping, read alignment, or read mapping after we have generated clean FASTQ files. Now, generation of this clean FASTQ files, as from the workflow you can see, it's a long process. So in this class, we'll go through this first. Okay. So first, before we go to individual details, first we want to like clear some concepts. So for the first one, it is sequencing depth versus coverage. What is sequencing depth? What is sequencing coverage? So basically, these are very similar, depth and coverage, in terms of usage, how we use. Like basically in papers also, you'll say they use depth and coverage, or like like once they use the term sequencing depth, sometimes they use the term sequencing coverage. These are like two very interchangeable terms that you'll see in literature as well. But the thing is, they actually mean uh, they're actually slightly different things, and they are basically very distinct things. But if you consider in terms of how uh, like what is the value of sequencing depth and coverage, they might be very similar. Why? You'll understand after these two slides. So sequencing depth, also known as the read depth or the depth of coverage, refers to the number of times a specific base or a nucleotide in the DNA is read during the sequencing process. So if you have a DNA, you have a stretch of read. So in that particular region, what is the number of times a particular base has been read? So a particular region of the DNA will be read multiple times, because that is how you are setting up your sequencing experiment. You are cutting your DNA, and then you are trying to like amplify using PCR and everything. So what happens is, most often, and that is how it is done, you'll find that two reads are overlapping by a lot of stretch of the DNA. So this number of overlaps, so if it is more than two reads, if it's like 5, 10, 20, 30 reads that are overlapping at a particular position of the genome on a particular base, that is the number is basically which gives you the sequencing depth or the depth of coverage. So it is basically the average number of times a given position in the genome it has been sequenced. So basically, how many reads have sequenced that particular position, that particular nucleotide. That is what sequencing depth gives you. So a higher sequencing depth provides more confidence in the accuracy of the base calls at that position, and it helps to reduce sequencing errors and noise. So intuitively thinking, if you think it like that, like 30 reads have sequenced that particular A. Let's assume it's an A in the genome there. So intuitively thinking, uh, 30 different reads have sequenced A at that particular position. So it gives you more confidence that that particular nucleotide there present there is of high confidence. Basically, your base caller has done a good job to call that particular base. So more number of times it has been sequenced by reads, the more evidence you have for that particular base to be the correct one, and more confidence you'll have in further downstream analysis. As I was saying, when you are checking for variants, like if that particular A has been changed to a T or a C or a G, how many reads are supporting that? That there comes another application, another importance of sequencing depth. There also, like how many reads does this variant support? That is a question that is often asked, and like if the number is higher, obviously you have high confidence in the particular variant existing. If the number of reads are lower, then obviously you have low confidence. The number specifies how many times that particular base has been sequenced. So if a specific nucleotide has been sequenced 30 times, the sequencing depth of that position is basically 30, given as 30x. It is written in that format, 30 and an x after that. So as I was saying, it refers to the number of times a particular nucleotide is read during the sequencing process. And what's the purpose of it? Higher sequencing depth increases the confidence in variant calling at specific location. It is particularly important when looking for rare variants or when sequencing heterogeneous samples like tumor tissues. And, and it is represented in that form, number and X depth. So this is what sequencing depth is. Now, if we go on to coverage, and so coverage is basically very closely related to sequencing depth, but it provides a much broader perspective. Coverage gives you the proportion or the percentage of the genome that has been sequenced at a certain depth. So it gives an idea of how much the entire genome has been effectively read, and is usually expressed as a multiple of the genome size. So, for example, the human genome is around 3 billion base pairs long on an average, and it is sequenced at an average depth of 30 times or 30x. The coverage would be 30x as well, because it is giving you on an average, each base on this genome has been sequenced 30 times. So you're assuming you've sequenced the whole genome, which is 3 billion base pairs long, and each of those base has been on an average sequenced 30 times. So what it what sequencing coverage tells you is basically the proportion of the genome that has been sequenced at least once. So the primary definition is at least once, and if your depth is given as a 30x or a 50x, you'll define it like that. Okay, like the whole genome uh, has been sequenced at a depth of 50x, and basically each of the base has been covered as 50 times. So on an average, your coverage will be 50x. So as you can see, the depth and the coverage are the numbers are exactly equal, because what you're assuming is the whole genome has been sequenced. If you assume in a situation like this, a particular region of this genome has a lot of depth, but the rest of the genome does not have that. So your average depth and your average coverage would be different. So there are various portions of the genome which is hard to sequence. So, for example, the repeat regions, which are very hard to sequence and align. So, in those cases, the number of the basically not all the repeat regions will be properly sequenced. So, your although your genome is like 3 billion base pairs long, so maybe just to give you an idea, so maybe you have sequenced only 2.8 or 2.9 billion base pairs, and rest of the zero point 0.1 or zero point 0.2 billion base pairs are repeats, so you could not sequence them and could not properly align them. So coverage will tell you if your whole genome has been sequenced or not, and if it has been sequenced, what is the average depth of the particular base that has been sequenced. So the concepts are very similar, sequencing depth and coverage, but coverage gives you a much more broader perspective, and it is very often used interchangeably, sequencing depth and coverage, but you should know what it means. So uh, what is the purpose of this metric sequencing coverage? What it gives you is it ensures that the entirety or as much as possible of the target region, whether it is the whole genome or the exome or a specific panel, has been sequenced. So it tells you if it is like it has been the whole genome has been sequenced or a part of the genome has been sequenced. Basically, whatever your target is, what percentage of it has been sequenced. Okay. Uh, and it is given as percentage, where basically 95% coverage means 95% of the intended region has been sequenced at least once. So in this definition, you will see like we are talking about at least once, but depending on your depth, you can change your definition. So if you want a depth of say 30x across the whole genome, so what you'll say in this case is that 95% of the genome has been covered at a depth of 30x, but rest of the genome has not been covered. So what are the factors that influence this coverage? Quality of the DNA sample, which is very obvious, library preparation, sequencing biases. There can be biases in your base calling software, which can introduce errors in your in the sequence generated. So sequencing biases and gaps in reference sequences can impact coverages as well. So it is possible in your in the sample that you've collected and you have sequenced the genome there, like you might have sequenced the whole genome, but the next issue that can arise is that maybe the whole genome of your reference is not well established. So there are parts of the genome in your reference file where the certain parts are missing. There are certain gaps in your reference file. So even though you have sequenced that part of your genome, that part you cannot claim uh, uh, like confidence in those parts because you do not have the reference on where will that part go and align. So there also your coverage will come down, not because of your sequencing issue, but because you do not have the proper reference and like gapless reference genome to align to. So these kind of issues might appear. And as I was saying, some regions of the genome may be hard to sequence due to high GC content or repetitive elements or any other genomic complexities. Mostly it's like repetitive elements, that's the issue. Uh, so as I was saying, I'll not go through it again and again. So both are important, depth and coverage. By increasing the sequencing depth, one could also boost the coverage because more reads mean a higher likelihood of covering more regions. This is intuitive. So if your number of reads is much more, you have a higher probability of covering the whole gene. So it, by increasing sequencing depth, you can increase the coverage as well. And you need to have a balance between high enough depth to detect variants confidently and a comprehensive coverage that it covers the whole genome to ensure that the entire target region is sequenced. And it can help you in variant calling and basically completing your whole data set. And you need to have a balance between the depth and the coverage. Okay. So this is a pictorial representation of what basically depth and breadth of coverage means. So depth of coverage is basically how many times a particular read has been sequenced. So say for example, this read here will have one, two, three, four, five reads supporting that particular base. Say for example, I have an A here. Sorry. I have an A here. So how many times this A has been sequenced? So it's basically 1, 2, 3, 4, 5. 5 reads have uh, have the evidence that there is an A at that position. So that is what is giving you the depth, and the breadth, or basically the sequencing coverage, is telling you how much of the genome has been sequenced. So per base coverage is the average number of times a base of a genome is sequenced. The coverage depth of a genome is calculated as the number of bases of all short reads that match a genome divided by the length of the genome. We'll come to this formula in the next slide. It is often expressed as 1x, 2x, or whatever number x. And as I was saying, breadth of coverage is basically how much of your genome is covered by those reads. Are there regions that are not covered even by a single read? So breadth of coverage is the percentage of bases of a reference genome uh, that are covered with a certain depth. So, for example, 90% of the genome might be covered at 1x depth, and 70%, still the 70% can be covered at 5x depth. So like the depth might not be uniform across your whole genome. So that's why we calculate what we call as mean, or mean depth or mean coverage. So it gives you an idea on an average how many times a particular base in the genome has been sequenced. And yeah, that's all. So this is the formula. Uh, if you look at it, so Lander and Waterman uh, describe this theoretical redundancy of coverage as ln by g, L into n divided by g, where L is the read length, n is the number of reads, and g is the haploid genome length. So haploid, basically, if you consider only one strand. So in the human genome is about 3 into 10 to the power 9, is 3 billion base pairs long. L will be like the average read length for that particular sequencing project. So for example, Illumina will have around read length 150 to 300 on that range. N will be the total number of reads that you have, and J is, and you divide the whole thing by the total length of your genome. So, biases in sample preparation, sequencing, and genomic alignment, and even assembly can result in regions of the genome that lack coverage, that is gaps. As I was discussing, gaps might be due to your reference, or maybe you have not sequenced that part, or whatever can be the reason. And and in regions with much higher coverage than theoretically explained. So basically, if you have, imagine this situation: you have a reference genome, and almost all of the genome has been sequenced at the depth of 5x, but at a certain position, say around like 500 base pairs, like in the middle of the genome, you have a sequencing depth of 50. Now, can somebody think and tell me what does this mean actually? The whole of the genome has been covered at a 5x, but at one certain portion of the genome, there has been 50x depth. Just give it a thought. What can that mean? Okay, so one explanation would be that might be that 500 base pair region has been duplicated in your own gene, in the genome that you're sequencing. So what happens is in that 500 base pair region, it, that is a 500 base pair region in your reference genome, but that is might be like a say 5000 base pair region where 500, 500, 500 has been repeated 10 times. So the reads that have generated from those regions, those repeat parts in your own genome, has the reads will be uh, the number of reads will be higher, because like it's the same read that has been generated multiple times due to duplication of the genome. So up that area of the reference genome, you'll have much higher depth, because a lot of more reads has been generated compared to the rest of the genome, because in your own genome compared to the one that in the reference, there has been a duplication of that region. That 500 base pair has been duplicated multiple times, so your number of reads has increased in that particular region, but rest of the region has a depth of like 5 or 6, and in that particular region it is like 50. So these are the kind of techniques that variant calling software is also used. So for example, when you're calling structural variations, or you're trying to call you're trying to see the single nucleotide variations inside your genome, you use techniques like this. Has your depth increased at that particular point, or has your depth decreased? If it is decreased, then one reason might be that it has been deleted. It is possible. So one or two reads have been generated, but your rest of the genome is in 10x. In that portion, only one or two reads are there. So it is possible that part of the that region has been deleted in your genome compared to the reference one. So these, like the coverage and the depth, they give you a lot more idea than you like initially think about. So they can give you the whole distribution of your genome, how your genome is distributed, how your different reads are distributed across your genome, and from that you can infer certain things. If there is a increase in uh, depth, there might be. Huh, as I was saying, if it is say for example six or seven x instead of 500x, it means that that portion of the genome has either not been sequenced properly, or there has been a deletion, say 500 base pair range, say maybe like 300 has been deleted. Now imagine Illumina has like 150 base pair range. So if a 300 portion of the read has been deleted, so that portion will have lower depth, because Illumina will not have a single read from that position, because 150 makes up 300, so two read position, it's gone. So you are not able to get the read for that particular region. So and the rest of the 500 base pair is there. So total 500, say for 100 has been deleted. Let's assume let's assume a bigger one. So 1000 base pair, say 300 has been deleted. So 150, 300 from the middle has been deleted, and it's a 1000 base pair. Now across a thousand, the whole the reads will be overlapping across all the thousand base pairs. Now in that particular small 100 base pair region, the number of reads will not be there. So what will happen is, in the whole thousand base pair region, your total number will go down, because you have not generated reads from certain portions of that region. So your total number will go down. So your sequencing depth will be going down. So if you have the whole sequencing, whole genome as like 500x, and your one part of the genome is showing 6 or 7x, what it means is around that region, certain things have been deleted. Certain part of the genome has been deleted, might have been deleted. Now, like this is an inference I'm making, but it can also be possible due to sequencing errors as well. You have to keep that in mind as well. So you have to check what is the quality of those particular reads as well. If when it is 6x or 7x compared to a 500x, what is the quality of those reads? We'll come to the quality a little bit later, but that is how you have to infer. Okay, is there a certain thing that has happened to the genome that is affecting my depth? If it is your sequencing error, you can fix it, but if it is not, uh, sorry, somebody raised a hand. I think. Okay. But mostly it will be some sort of variation in the gene. So as I was saying, if it is duplicated, if you like a single portion has been taken multiple times, so there will be multiple reads producing from that portion of the genome. But when you're trying to align it to the reference genome, that is only a small part that has not been repeated. So on below of that, your reads will stack up. So your sequencing depth will increase. Yeah. Anyone has a question? Okay, we'll move on. Okay. So uh, one such example, other than like a variation in the genome, one such example would be GC rich regions, which are known as CPG islands. These are basically particularly prone to low depth of coverage, partly because these regions remain anneal during amplification, and these regions are not well amplified. So if you recall, GC is okay. So basically, if you have a genome, say for example, you have a 1000 base pair genome, a middle part has been deleted out. So if it has been deleted out, how can you sequence that? If if that part has been deleted, you cannot sequence that, and you'll see that part has been deleted because when you're trying to align your sequence read to your reference genome, no reads will go and align to that part of the genome where that part has been deleted. So you can assume that part has been deleted because no reads are there present at that position in the genome. So in your genome, it has been deleted, but in the reference genome, it's still there. So comparing your reference genome to whatever reads that you have got, you can claim that it might have been deleted because there are no reads originating from that portion of the genome in your sample. But in your reference, it's there. But in your sample, you have no reads corresponding to that portion of the genome. Then you can claim that this might have been deleted, right? Okay, moving on. So this GC rich regions, so the issue there is basically this uh, GC are basically, if you remember from uh, like elementary molecular biology, so GC forms triple hydrogen bonds compared to AT, right? So if you have a GC rich region, what it does is it like turns on itself and forms this double bonds. So a single stranded DNA will be formed into a double stranded DNA because GC are very they're very attracted to each other because they're forming triple hydrogen bonds, and those are very strong bonds. So when you are doing amplification using PCR during sequencing, the basically those part of the region which has high GC content, those will not be amplified that much because it the single strand will not be there, so it will come back and bind on itself, form a double strand. So what how is PCR done? On a single strand, you give up DNA polymerase and it generates another strand. But in this case, the single strand is already a double strand because of the GC rich region. So the amplification process is not done properly. So the number of reads arising from this GC rich region is very low because of your inability to amplify those regions properly. So this is one of the examples where you have low reads, but it's not due to deletion or it's not due to any kind of variation, but it's mostly due to GC rich regions. Because you not have been able to amplify this properly. So these are like sequencing issues that come up, uh, and that can give you low depth or low coverage. But you need to have the intuition to identify the difference, which is coming from a sequencing error and which is coming because actually a variant is present in there. So we'll cover this going on, but I just want to give you an idea. So yeah, we've covered mostly everything in terms of depth and coverage. So I just wanted to give you an idea about how different coverages look. So it is a black line, you see. This is basically human genome at 1x coverage. So basically, this is giving you a number of reads, and this is the read length. So y-axis is your number of reads, and x-axis is your read length. So if you see, this is the 1x line. Basically, if you sequence at least sequence each of the base pairs of the nucleotides at least one time. So this is the line that is showing that. So you on an average have around 10 to the power. Uh, I'm not being able to see the number, but I'm guessing this is 10 to the power 8. So around 10 to the power 8, and your read length will vary depending on your basically the sequencing protocol. So according to that formula that I showed, ln by g, read length into number of leads by divided by the length of the genome. This line gives you the coverage basically, but the actual coverage that we get as an outcome from the different sequences that we use are basically quite high. If you can see, Illumina HiSeq gives you quite high coverage because the number of feeds is very high compared to the read length. So these kind of new technologies that we have, Illumina and different ion torrent, uh, these three are Illumina technologies. So basically, PacBio as well. PacBio, if you remember, it's a long read data, so the read length will be higher, so number of reads will be a little bit lower. So Illumina are short read, so your read length will be little around like this is around the 150 mark, and your number of reads will be higher. So you can like think of that intuitively as well. So if your individual read length becomes higher, you need less number of reads to cover the full genome. So this is just to give you an idea about how different types of sequences look like. So this is the line for 30x coverage. So you see HiSeq 2000. This is a very widely used sequencer gives you more than 30x coverage. So for a WGS data set, so WGS is basically whole genome sequencing. If for a whole genome sequencing data set, 30x is a decent amount of coverage to have, because like 3 billion base pairs, each base pair you have covered at least 30 times. So that's a lot. That is good enough to be a good data. So yeah, and this is basically a plot that is showing you different types of experiments and their requirement for no, sorry uh, and they require different types of basically uh, read length requirement. So sorry, sequencing depth requirement. So basically, this is giving you a number of bases sequenced per sample, and this is giving you the frequency of studies. So if you see, WES is like in the middle. Like if you have uh, W is basically whole exome sequencing. So you have exome sequencing on the left, you have ChIP-seq, on the right, you have whole genome sequencing. So for whole genome sequencing, you would want a little bit more coverage because you're sequencing the whole genome. For exome, you are only sequencing targeted portions of the genome, the regions only which code for protein. So in those cases, you have some prior knowledge, so a little bit of less depth can be used. And for ChIP-seq, will not go into ChIP-seq and RNA-seq, but in those cases, a little lower depth is acceptable as well. But for whole genome, the range is like around 30x to 50x. That is the standard range. If you see different papers, Illumina, for short read, it's Illumina, you see around 30x to 50x coverage and depth as well. So yeah. So next we come to the part of like our sequencer has provided us with some sort of signal, be it fluorescence, be it radioactivity, be it any other kind of signal. Now, how do we take that signal and convert it to a base? Basically, not a base. Basically, what we do is we try and identify the signal and identify which nucleotide actually caused that signal. So, in this step, we'll learn how to do that. So, uh, sequencing quality metrics basically how good is the quality of the base that you have identified. Basically, how confident are you that at that position that there is that base only, because the sequencer doesn't give you an readout key like it's like ATGC, ATGC, something like that. What it gives you as a readout is basically signals. Say at that position, this is the signal I got. At this position, this is the signal I got. Now you need to convert those signals into actual meaningful data, basically the sequence of nucleotides. So the base caller does that, but it is not error free, as you can imagine. Like, there always will be error associated with this type of technology. So it's not exactly error free. There will be errors at certain positions, but how do you quantify that? Using something called a quality score or a Fred quality score. So sequencing matrix can provide important information about the accuracy of each step in this process, including library preparation, base calling, read alignment, and variant calling. Base calling accuracy, it is measured by this Fred quality score. It is the most common metric used to assess the accuracy of a sequencing platform. It indicates the probability that a given base is incorrectly called by the sequencer. So the quality score is basically giving you a probabilistic value. Okay, like this is the probability that the sequencer has called this base incorrectly, or the base caller has identified this signal incorrectly and assigned an incorrect base to this signal. So this quality score is basically was historically used to determine Sanger sequencing accuracy. It's originated as an algorithmic approach to consider Sanger sequencing metrics such as peak resolution and shape. And as you remember, Sanger sequencing gave us an output of uh, basically what we call a chromatogram, and how were the different peaks of the signals uh, that is coming out from the sequencer. We'll see one, so I'll just move forward. So this is how a chromatogram looks, as you all know. So each of the signal corresponding to a particular base. So uh, yeah, and link them to the known sequence accuracy through large multivariate lookup tables. So there are lookup tables. If you have this kind of signal, the lookup table will tell you it might be this base with a certain confidence or a certain probability. It's not always 100% accurate. It'll give you a it'll give you an output, but with a certain probability. it being correct only. Okay. This method proved to be highly accurate across a range of sequencing chemistries and instruments. So then it was after Sanger sequencing, it was adopted in actually almost all kind of sequencing technologies. So you'll have that Fred's quality scoring, which will give you the accuracy of the base call. So parameters relevant to a particular sequencing chemistry are analyzed for a large empirical data set of known accuracy. The resulting quality score lookup tables are used to calculate quality score for de novo next generation sequencing data and possessing an equivalent meaning to the historical metrics familiar to the most sequencing Sanger sequencing users. So Fed scale, this is basically what the quality scores gives you. In the context of sequencing, the Fred scaled quality scores are used to represent how confident we are in the assignment of each base by a sequencer. In the context of variant calling, Fred quality scores can be used to represent many types of probabilities. So you can have quality scores at the level of base calling, basically where your base caller is identifying a signal and assigning them a particular nucleotide. You can have a quality score there. You can also have a quality score in terms of variant calling when you're using variant calling softwares and identifying different types of variants in your genome. You can use this Fred quality scores to represent this different probabilities. GATK is a very commonly used software. If you just look it up, it is you can see it. We'll just show you here. So, oh, sorry, this is the GitHub page. This is the whole toolkit that has been developed by Broad Institute for all kind of genome analysis that you can think of. So this is known as GATK, Genome Analysis Toolkit. I'll leave the link here if anybody is interested. Okay, moving on. So the most commonly used in GATK is the qual score or the variant quality score. It is used in much the same way as the base quality score, the Fred quality score that we saw for the base calling. And the variant quality score is a Fred scaled estimate of how confident we are that the variant caller correctly identified that a given genome position displays variation at least in one cell. So in practice, by convention, most usable Fred scaled base quality scores range from around 2 to 40. That's the normal range, with some variations in the range depending on the origin of the sequencing data. However, Fred scaled quality scores in general can range from anywhere from 0 to infinity, theoretically speaking. A higher score indicates a higher probability that a particular decision is correct, that a particular base calling is correct. Basically, the particular assignment of nucleotide at that position is correct if you have a higher Fred score. And conversely, a lower score will indicate that a higher probability that the decision might be incorrect. And if you assume the error probability to be E, your quality score or the Fred quality score is given as minus of log 10. Sorry, 10 log E. Yeah. So your E is the probability of the error and your Q here is the Fred quality score. Now, uh, we can interpret this score as an estimate of error where the error is the probability that the base has been called inaccurately by the sequencer, but we also can interpret as an estimate of accuracy. The accuracy is the probability that the base identifier has called it correctly. Depending on how we decide to express it, we can make certain modifications to this. So like you just take what you do is basically take the 10 here and take an anti-log here. So this is what will give you the value of E. It's simple. And if you want to, this is basically the probability of error. Now, if you want to check the probability of accuracy, just do a 1 minus E because this is a probability value. The total probability is 1. So 1 minus error will be your accuracy. So this is a table which is showing you how different Fred quality, what different Fred quality mean in terms of error and accuracy. And in general, a Fred score of at least 20 and above is acceptable because a Fred score of 20 means whatever it qualifies is actually 99% accurate and with one chance of error, 1% chance of error. So if you see Fred quality score of 10, if you check like the error rate here if you put a 10 in q, so it is becoming 10 to the power minus 1, which is basically 1 by 10. It's basically 10% error and 90% accurate. So 1 minus 10 will be 90. Now, if you do like a higher Phred score, like 20, so if you substitute 20 in here, so it's basically 10 to the power minus 2, it becomes. So it's basically 1 by 100, which is 1% of error and 99% accurate. So this is something that is acceptable around 20 Phred score, or basically the quality score. So 20 is generally what is the cutoff for acceptable quality scores, which is actually giving you 1% error and 99% accuracy. And if you increase, it'll go from 99 to 99.9, 99.99, and how many nines you want to add, depending if you go on increasing the Phred score. But around 20 and 30 is something that we generally look for. Okay, now we learned about different uh, quality scores, how we calculate quality scores for the base callers. Now we'll look at different type of output formats for different type of sequencers. So first is the oldest one, Sanger output format. Excuse me. So the output of the Sanger sequencing, if you recall, Sanger sequencing will give you basically uh, in this case, it's basically a chromatogram which tells you, okay, like what kind of fluorescence or what kind of light has been emitted for a particular base. So the output of the Sanger sequencing is typically a chromatogram and also known as a trace. So instead of a chromatogram, it is also called a trace, and basically a text-based sequence file. Although it may seem to hold all relevant information, after all, the point of sequencing is get to sequence, but like converting the chromatogram to the text-based sequence file, that is something that is done by the base callers. So uh, so the chromatogram shown by most trace viewers is a processed version. Raw data from the sequencer is manipulated by analysis software to facilitate base calling. The start of the process trace, assigned position 1, is the software's best case of the first peak. So basically, this is how the chromatogram is looking like. Your y-axis has your signal intensity and your x-axis has your time. So it is given in relative fluorescence unit because the signal is basically fluorescence, and it's plotted over time, which correlates with the base position. Now on the top you can see these are the bases that are assigned, and these are the fluorescence peaks. Analysis software processes the raw data and assigns bases to the peaks to generate the final trace and a sequence. Quality values are calculated for each base. When you're trying to assign that base, you'll have a quality score associated with it, which is given by these dashes on the top. And quality values are calculated for each assigned base, providing a measure of the confidence of the base call, as we were checking Phred quality score. It's quite simple. So for the first 20 to 40 base pairs, you'll notice that it is not very well resolved. So if you see on the lower end, your peaks are very distinct. So this peak is there, your next peak is actually in the trough of this curve. So your peaks are very distinct in terms of ATGC. So these are like each of the curve is a different color of curve is showing you different basically bases or nucleotides. So if your peaks are all together at once, say for example this one at this position, all the peaks are together. So what will the base caller say? Is it ATG or C? Like it can be anything according to the peaks. But on the later stages, if you see, the peaks are quite well resolved. So at this position, there is this peak, and the next position there is a separate peak. Not like this. This is all messed up at the beginning. So the first 20 to 40 base pairs are typically not well resolved. Very short sequencing products do not migrate in predictable manner during capillary electrophoresis. If you remember, Sanger sequencing used electrophoresis to like separate out the different DNA fragments. And the analysis software has difficulty assigning bases within this region, causing the ends to appear in this region. So basically, if the base callers doesn't assign a particular base to this to a particular region of the DNA or the genome, what it'll do is it will replace your ATGC with Ns. So what N means is basically you do not know what the base is there. So, but you know that there is a base there. So you want to show that there is certain amount of this genome which you have not been able to sequence. So you just put an Ns there. That is what N means. And uh, to avoid critical data fall in this region, it is recommended designing primers that can that can bind at least 60 base pairs, preferably 100 base pairs away. So, like if you have a leverage of 100 base pairs in the beginning, so let it call out all the wrong callers at first. Then after 40 or 50 or 60 base pairs, you'll get the right calls because typically, like on the lower end, it's not very well resolved. Note that the sequence of the primer will not be seen in the results as bases within the primers are not labeled during expansion reaction. So you need to be a little bit careful when you are designing primers like this. So you need to be careful and mark those primers to like what will happen is after the end of sequencing, you need to identify which are the adapters and which is your actual DNA, actual sample genome. So you need to be a little bit more careful in this. Now, that was the start of the chromatogram. Now, in the middle, most sequencing protocols are optimized to provide the best peak resolution, roughly between 100 and 500 base pairs. In this range, the peaks should be sharp and well spaced. The base calling is most reliable. You see like how good the peaks are, like at each one. So, if there is a peak of green, there is no other color to be seen. All are like almost zero. But if you see here, sorry, if you see here, it's like all of them are at once. Still, these are like better, but if you see here, this is like much better. If you have a peak of one color, you do not have any kind of other colors at only. So typically between 100 and 500 bases, there is where your peaks get resolved very well, and that is the region where you are the most confident in um, base calling, and it's the most reliable part of the genome. And towards the end of the trace, again, towards the end of the chromatogram, uh, expect peaks to be less defined and lower in intensity. The base calling will be also be less reliable due to the nature of in vitro polymerization. Large sequencing products are generated less efficiently than their shorter counterparts. So the more you basically increase your read length, the more these peaks will come together and like resolution will be much more difficult. So the larger products are fewer in number and produce a weaker signal. And also with electrophoresis method, it becomes increasingly difficult to resolve a single base difference as DNA fragments become larger. So if you remember last class also we discussed the Sanger, one of the drawbacks of Sanger sequencing is like it can detect when you are doing an electrophoresis assay after basically using small sequences of DNA and synthesizing it. When you're doing this assay, it is easier to resolve a base pair with like resolve two segments of length 20 and 30. So if you have a 20 base pair length segment and you have another 30 base pair sequence. Now these might resolve quite well in your electrophoresis. But if you increase your length, if you move up to say 100 base pair, so 100 and 101, it's harder to resolve, and and 1000 and 1001 base pair will be more harder to resolve. So your peaks will come starting cluttering together, and you will not have good peaks as you saw in Lennon the 300 base pair range. You will not have good peaks. So at the end also you'll find a similar situation at the beginning. It's not well resolved, and you have a cluster of peaks together. So you are most confident in this region, 100 to 500 base pair region, because the peaks are very good, and uh, like two or three different peaks do not interfere with each other. So it's the most reliable calling that is coming from the middle of the chromatogram. Now, that we have understood in general what are quality scores, what are different, like how do different base callers like identify the different nucleotides at particular regions. Now what we'll see is how we put them in different data formats. So the most simplest of these data formats are the FASTA format. So FASTA is a simple, lean format which is used to represent longer DNA sequences that have been typically established from a lot of sequencing and which no longer travel with their quality scores. So you do not have quality scores associated with FASTA. So you will typically put those DNA in those genome in your FASTA region, which are basically 100% confident that it's the 100% confident correct sequence. So in most cases, this will be your reference genomes because reference genomes is something that you consider as a truth set and you compare your sample genome with those. So in the FASTA format with no quality scores assigned, mostly what you'll put in are reference genomes. Uh, this is a FASTA format which we will typically see storing the DNA sequence from their reference units. FASTA files typically use file extensions. You'll have a .fa or a .fasta or a .fna. .fna means basically it's nucleotide. You can also have a FASTA sequence of proteins, like single letter proteins written one after, single letter amino acids written one after another. But in, like most cases, if it's not a protein, it's mostly nucleotides. So it's either a .fa or a .fna. So in an ideal world, a reference genome would contain a single uninterrupted sequence of DNA for every chromosome. While resources for some well-studied species include chromosomal level assemblies, which have much sequence organized into the chromosomes in a FASTA file, even these genome assemblies often include a large number of short fragments of sequence that are known to belong to the species, but whose location and at which location and orientation in the genome remains unknown. So as I was saying, like if you remember, go back to that scenario, if your part of your genome has been deleted, a part of the genome has been duplicated. So what you'll see is you'll have the reads, but you'll not know from where did these reads came from. So it's a similar situation here. So in even in big assemblies, what you'll find are shorter regions which you you know that these regions shorter stretches of DNA. So you know that these reads have come from this particular species only, but you do not know where and from uh, the location and the orientation. Both you don't know. So the reference genome for an organism you are working on might be the product of a recent small-scale assembly of a low coverage genome. In this case, the genome may be represented by thousands or 10,000s of scaffolds, and only a few might be longer than one or few megabases. All of these scaffolds go into the FASTA file of the reference genome. So this is how the FASTA file looks like. So I'll go into the detail of each line, what it means, what these different formats mean. But from the second line, if you ignore the first line for a moment, from the second line, what you see is basically the stretch of ATGC, ATGC. Now you'll notice some ATGC are small in letter, some are capital, and if you look very carefully, I cannot find it, but there will be certain N's as well. As I was saying, if the base caller is not confident what base is that, it'll put an N. So it's a very straightforward format. So any line starting with this greater than arrow sign is represented as a line holding the identifier of the following sequence. So if this is the sequence, the first line starting with this greater than arrow will tell you the identifier of the sequence. What does the identifier is? So basically here it is this number which is encoded by anything, like this might be your sample number, or this might be your whatever kind of encoding you're doing with this sample, you can put a number here. The remainder of the line, following the first white space, are further comments about the sequence, but not typically be carried through downstream analysis, such as alignment pipelines. In this case, so this is the identifier, is the name of the assembled chromosome in this reference genome. The remaining lines give DNA sequence of that assembled chromosome. Uh, so this is basically what information is giving you is this is basically telling you the genus and the species, basically the scientific name of the organism you have sequenced. These are other details about the sequencing method. So this is maybe an isolated something they have isolated, some part of the genome they have isolated from this organism. Uh, that has been sequenced and that uh, sequence is here, and maybe this is some other extra information that you have added. So in that line, the information that you are adding might should be in the first line only, and after the first line, from your second line, your whole sequence the sequence will start without any gaps. Okay, so in this fragment, you see DNA sequence that is either in lowercase or in uppercase. A common convention is that the lowercase of the DNA have been inferred to include repetitive DNA. So what they do is, so a part of the genome, if it has been like translated from a like a caps letter to a small letter, what it means is those part of the genome are showing repetition. So if you see this part, so T like you have around 5 A's like this. So this part of the genome has been repeated. So that's why what they do is, this is what is called as soft masking. You convert the capital letters into a small letter so that these sequence don't go and uh, like erroneously aligned to different parts of the genome. You know these are repeat regions, so you're already telling your alignment software, please consider them separately because these are repeat regions and these are repeat sequences. Do not consider them to be the same as the rest of the sequence. This is the information you're trying to provide, and you can do this thing using softwares like Repeat Master. But what basically the phenomenon is called as soft masking. This is how we mask the data. But just to remember, not all reference genome will have these repeat sequences denoted in this manner. Some will like just straight up be the same across all. So you're not giving that information to your alignment software that this part is repeated. It does whatever it does. Sometimes it will be deleted off, sometimes it will be substituted with n. So if it is substituted with n, what you're trying to say is, I know this is a problematic region of the genome and I do not want any kind of alignment with using this part. So you completely shut off that part of the genome, so that other erroneous reads do not come and bind to the part. So you can do like multiple things for masking. So if you're using n instead of the capital letters, it's basically hard masking, and if you just convert the capital to a small letter, it's basically soft masking. And as I was saying, most reference genome contain gaps. Sometimes the length of these gaps can be accurately known, in which case each missing base pair is replaced by n. As I was saying, some gaps of unknown length are represented by a string of n with some fixed length, like 100. So you can do these tweaks depending on the genome. Oh, sorry. Like, can you see the PPT now? Sorry, I thought it was sharing. Can you see it now?
Yes, sir.
Okay. Please let me know if you're not able to see, because from my end, it's not possible for me to know if you're not able to see or not. So, like, I'll keep on talking and you'll not be able to see. So please let me know next time when you're not able to see by unmuting yourself. Okay. Okay. So this was the FASTA format. So few key things to keep in mind. It's just showing you the sequence of your nucleotides in the genome. On the top, you have an identifier. Along with that, you have certain other information added to it. What species is it coming from? What kind of chemical treatment you have done it with it? Maybe some part of it you have isolated, and uh, that sequence is what it is representing here. So these kind of information you put it in the first line, and from the next line you have all the sequences. And in those sequences, you can do masking or no masking depending upon your requirement. But this is how the format looks like. But the most important thing to remember here is this format does not have a quality score associated with it. So, for example, you know that this is the sequence of the genome. This is the whole sequence of the genome, but you do not know the quality scores associated with the individual nucleotides. You do not know if the second G here is actually a confident base call or an or a, like a erroneous base call. You do not have any idea about that. So FASTA format will only use for sequences that we are 100% confident of. In most cases, these are reference genomes, as I was saying, but the quality scores are not associated here in the FASTA format. So in the Illumina outcome that we get, what we call is as FASTQ format. It's very similar to the FASTA format, but along with that, you have certain other information as well, like the quality scores and other information. I'll come to that. But basically, FASTQ format is the standard format for data coming out of an Illumina machine. So all Illumina data you'll get in FASTQ format. Even other sequences also you can get data in FASTQ formats. This is one of the most widely used formats in the omics space. So as we have seen, paired end sequencing produces two separate reads of the DNA fragment. If you remember Illumina, it sequences from both ends of a read. So it sequences from 5' to 3'. It also sequences from 3' to 5'. So basically you have, for the particular read, you have two readouts. So particular region of the genome, this is the read. You have two reads from this end, from this end as well. So, paired end sequencing produces two separate reads of DNA fragment. Those two different reads are usually stored in separate files named in such a manner as to transparently denote whether it contains sequences obtained from read 1 and read 2. So, what will happen is, from the sequence readout file, you'll have two FASTQ files. One FASTQ file for the read 1 and the second FASTQ file for the read 2. But, say for example, the first line of your read 1 FASTQ file should correspond to the first line of your read to FASTQ file. So basically, it's if there are two FASTQ files, if you are reading say R1 in this direction, in basically 5' to 3' direction, in the first line of your FASTQ file, you have a stretch of 100 base pairs, say for example, and you're reading from 5' to 3'. So in the first FASTQ file, you'll have this sequence. The second FASTQ file, in the first line only, in the same position only, you'll have the sequence that you have read from the opposite direction. So this orientation is important because this is how the data structure is read. So you have to be careful. Okay, your read 1 and read 2 line should match in both the files. The files will be separate, but they should match in terms of, like, this is the sequence for read 1 from this direction, and this is the sequence from read 2 from the opposite direction. But uh, like they are in a separate file, but the line number is the same. The first line, it's denoting the first read, and here in the first line also it is denoting the first read in the opposite direction. So that you have to keep in mind. The FASTQ format is very simple. Information about each read occupies just 4 lines. This means that the number of lines in a proper FASTQ file must always be a multiple of 4. Something if you're interested in, just a small caveat. Okay. Briefly, the 4 lines of information about each read are always in the same order as follows. So you have an identifier line. So in this case, this was the FASTA file, this was the identifier line. In a similar way, you'll have an identifier line for your FASTQ file. Then you'll have the DNA sequence as it was there in a FASTA file. But after that, uh, the line, the third line will always almost simply start with a positive sign, but it can be optionally followed by a repeat of the ID line. That is something that you can encode depending upon what you want to put in there. And after that, there will be an ASCII coded Fred scale based quality score. So remember, in FASTA, we are not getting quality scores, but in FASTQ, we are incorporating quality scores for each of the bases. This will give us an estimated measure of certainty about each of the base call in the sequence, because you'll have an associated quality score associated with it. So this is how a FASTQ file looks like. So this is your identifier here. So these are different things that are denoted here. So for example, this is like denoting lane 1. This is denoting some, this is the sample ID maybe. This is the length of the sequence that is getting generated. So you have, this is your basically DNA sequence, most importantly. Next is followed by a plus sign. But this line is a very important line. So this line, what it is telling you are, these are coded values of quality scores. So basically uh, there is something, so basically if it is given A here, it's basically encoded by some quality score. So basically, if your quality score is 20, you are putting A there. If your quality score is 30, maybe you are putting F there. So this kind of encoding works in ASCII. This is basically in ASCII format. So it's an encoding of Fred plus 33. So whatever the Fred score is there, you add a 33 on top of that, and then you encode it with a ASCII character. So these are basically all encoded quality score values, but these are all representing some sort of quality values. So this is how the FASTQ file looks like. It's slightly different in terms of the last line, which is basically giving you the fourth line, basically. Sorry, the fourth line, which is basically giving you the quality scores of each base in your sequence. Okay, and after this plus sign, also you can put certain information if you want. But yeah, this is the standard format how it looks like. So as I was saying, the identifier line can just be about any string that starts with an at the rate sign. But from intermediate data, you might see something like this. Basically, different, like you have an identifier, then you have put a colon, then you put another identifier, then you put a colon. So depending upon what what information you want to put in the first line, you can put it. These are all IDs. So the colons and these are like basically field separators, delimiters. The separate parts of the line are above are interpreted something along the lines as follows. So your at the rate is your mandatory character that starts with the ID line. So basically, if you put an at the rate, the reader will identify that as an ID line. So it will not read that as a sequence. It will read the line after that as a sequence. So the next number here is basically giving the unique sequencing machine ID. You can encode that. 64 is basically the run number on the instrument, how many runs it has gone through, and which run. Yeah, the data from which run is present in this FASTQ file. So in this case, it's 64. The next ID is basically the flow cell identifier. So basically, if you remember, there are during Illumina sequencing, you have these flow cells, small flow cells, inside which there are these oligos attached, on which these adapters go and attach, and you can do DNA polymerase and those stuff. So this sequencing occurs in these small stretches of lanes inside the flow cell. So uh, this is the flow cell identifier, and one is giving you the lane number, at which lane has this been sequenced. Then you have the tile number and also the x coordinate and the y coordinate of the cluster of the tile, and whether it is read 1 or read 2 in the final one. So if this is coming from read 1. So these kind of information you can put in the first line of the pass file, followed by this line will be the sequence, and after that there will be a plus sign. You can put some more information here. After that, this is the most important line, which will give you the quality scores of these species called. Okay, so as I was saying, the fourth line will basically give you the quality score. It'll give you an estimate of the probability that the base call is correct or incorrect. Uh, this is given by this minus log 10 of the p-value, where p is the probability that the base is got incorrectly called, and the q value is basically called the Fred scale base quality score, as we were discussing. Yeah, Komal. So basically, in this live session, we revise all the things that has been taught in that particular week, and after that, towards the end, we solve some assignment problems as well. So it's basically giving you a revision and also solving some certain problems. Sorry. Okay. Yeah, so as I was saying, in the last or the fourth line, you'll get this ASCII coded characters, which will tell you about the quality scores of the different pieces that is present in the sequence. So I'll not go over all this again. We talked about this earlier, but yeah, uh, if you see like the score line, it's something like this, like it's weird characters one after another. Basically what it gives is from a file storage and parsing perspective, it makes sense to only use a single character to store the quality score for each base. So uh, that has been done. Each of those single characters represent a quality score, a number between 0 to 40. It's basically a ASCII form of encoding. So this is basically the Illumina page for where they have explained the FASTQ files and everything. You can go and take a look if you are interested. I'll put the link here. But yeah, it'll tell you all the details of how do you use FastQ, how you read them, what are the different softwares you can use to convert from different data formats to FastQ and vice versa and everything. If you're interested, you can try that out. Okay, so now comes the question of like so different type of sequences we saw last time. This time we're seeing different type of data formats coming from sequencing platforms. But the question remains, like what is that you are going to use? So, for example, if you're trying to answer a question, what type of technology you should use? Should you sequence the genome? Should you sequence the mRNAs? Should you sequence the proteins? Should you sequence something else, some metabolites or anything else? So, what are you trying to answer is the most important thing. So depending on that, you have this like the big four omics is basically genomics, transcriptomics, proteomics, and metabolomics. From the names, it's quite clear. Genomics deals with the genomic data, basically coming directly from your DNA. Transcriptomics deals with the mRNA data from basically your mRNA and your gene expression data. And proteomics deals with your protein structures and protein expression data. And metabolomics primarily deals with metabolites. So different kinds of metabolites, how they are uh, basically what their structure is, what is their concentration across different conditions and everything. So these are the big four omics, depending on what you're trying to do. Other than that, there is something called apiomics. So apiomics is, you'll not hear the term apiomics much, but you'll hear epigenomics or epitranscriptomics or epiproteomics. So what happens is basically uh, uh, like two types of technologies are used. So one, either you can sequence the whole thing, uh, or you can do mass spectrometry. Mass spectrometry will give you it's not an NGS sequencing platform. It's basically another type of technology which will give you what is present after what. Say for example, your sequence you want to know the sequence of a protein. So if you do an MS spectrometry, you'll get what are the amino acids that are forming up this protein. So on the left are the stuff that are sequencing. On the right, which are the stuff that you can do with mass spectrometry. It's basically mass spectrometry. How is it different from sequencing? It's basically mass spectrometry will break down your whole maybe the protein or your uh, mRNA or metabolite, whatever it is, it will break down into its individual components, and according to mass, so what will give you is the mass of each component of the of your whole compound. So according to the mass, you can then compute what element was there. So according to the molecular mass, you can say okay, this might have been a leucine, this might have been a isoleucine, or whatever amino acid is there. So depending on whatever question you're trying to answer, there are different types of technologies which use both sequencing and mass spectrometry. So this course we are mostly focused on sequencing, but I wanted to give you an idea. Okay, like they are not very, what to say, exclusive of each other. So depending on what you're trying to answer, you can do some sequencing experiments, you can do some mass spectrometry experiments, and you can combine both of them to get a result, or you can do either or depending on whatever question you're trying to answer. But the possibilities are huge. The possibilities are endless. You have all these technologies available right now. So you, depending on the question you're trying to answer, you can do a lot of stuff. So yes, this slide was just to give you an overview about how different types of technologies are there for different types of molecules inside the cell. So for proteins, it's proteomics. For mRNA, it's transcriptomics. For genes, it's genomics. For metabolic, it's metabolomics. If you are looking at epigenomic epigenomic modifications, for example, all of you, I guess, might know. Okay, The DNA is not in an eukaryotic system. The DNA is not actually. So, yeah, these sessions are recorded, and this will be posted in YouTube as well after the class. You can search it by my name. My YouTube channel will come up, and you can check these videos out later as well. Okay, then moving on. Now we have the data. We saw how data is getting generated from your basically sequencing machines, how they are base calling. Now you have the data. You either put it in FASTA, whatever you put it in, you have the raw data. Now you have to do certain quality control on the raw data, because obviously there will be some errors associated with the raw data. So you need to do some sort of quality control. So eh, before doing any kind of downstream analysis like variant calling or anything like that, you need to check certain things in your data. First is the obvious one, checking the quality of the base calls to ensure there are no issues during sequencing. Next is examining the reads to ensure that the quality metrics adhere to our expectations for experiment. So it is possible you're expecting some low quality reads, but your quality is through the roof. That might mean that there's some issue with your experiment as well. So it might be revisit that experiment or try it in a different way. And also there will be contamination in your reads as well. So like not the all of the reads will come from your genome. Some will be like improperly cut, some will be improperly sequenced, some will be mitochondrial DNA that you don't want to sequence, but it is coming in your sample. There can be other kinds of contamination as well. So the reads will not always be clean. You need to check for contamination and remove if there are any. So, for example, mitochondrial reads. You do not want mitochondrial reads. You want just the nuclear reads. So you have to find and remove these mitochondrial reads from your data. So the raw QC goals include identifying sequencing problems and determining whether there is a need to contact the sequencing facility, identifying overrepresented contaminating sequences, gaining insight into library complexity, and ensuring the organism is properly represented by the GC percent. So this is a metric that we measure for actually all chromosomes, and it is quite distinct for different organisms. It's the GC content. So your GC content, basically guanine and cytosine, how much of your whole genome is basically just G and C, these two only. So it should be ideally around like 50 to 60%, but sometimes very high, sometimes it's very low. So which will represent that you have some issue with your data. So that this is also some another important metric that we check for, to check if there is contamination or if there is any data quality issues. Okay. So FastQC is one such software that you can use to check the quality of the data. It provides a simple way to do some quality control checks on raw sequence data. So the main functions is basically you can import data from FastQ files. Now you can also use BAM and some alignment files as well, and it will give you an result in an HTML based report. And this is how the results look like. So this is the header file. Basically, this is basically the first module, which will give you the basic statistics of the sample, how many sequences are there, if there are any sequence flagged as poor quality, what is the sequence length, what is the percent GC content, and like all these like very basic statistics they'll give you in the first part. In the second part, you'll have this plot, which is basically per base sequence quality. So this is your position in the read, and this basically box plot will tell you the sequence quality of in that position, the sequence quality of the reads in that position. So and this red line will tell you the median quality. Okay, the median is basically the 50th percentile quality. So uh, this plot can tell us whether there are any problems occurring during sequencing, or whether we might need to contact the sequencing facility as well. So if you see the reads, these are the first few reads. The first few reads are okay, it's in the range of like 30 to 34. But as you move on, sequence quality becomes good, but then it starts to drop. So now you have a lot of reads that have a mean of less than 30, and you have a lot of big error bars, meaning like you have one read say having a quality of like 5 and another read having quality of 40. So the error bars are also very high. So you do not have very good sequencing quality data in according to this chart. So like if you see something like this, you can understand your read quality is not good. Other things that it provides is basically the per sequence. Yeah, okay. Yeah, this is the per sequence quality scores. So it, this is the average around this 35, 36 is the average quality per read. So you have around 30, the most of the reads having around this number. So you have a few reads which have like around 12, 10 quality scores, but the maximum of the reads you have a high quality score. So which is telling you that you have a good data set. The next thing that you check for is the per sequence GC content, which gives you GC distribution over all sequences. Generally, it's a good idea to note whether the GC content of the central peak corresponds to the expected GC percentage of the organism, because every organism will have an expected GC percentage, and it is different for different organisms. So it is organism specific, just to be clear. And the distribution should look normal. So this is a normal distribution, as you can see, a Gaussian distribution. This is how theoretically it should be present, but in your real life data, basically the red line, you can see the GC content is like quite spread over, and which can tell you that there might be a contamination with another organism. So this is possible. And this would plot would indicate some type of overrepresented sequences with sharp peaks indicating either the contamination or a highly overexpressed gene. Uh, okay, okay. Moving on. Next, what plots it will generate is basically, this is basically telling you sequence duplication levels. You can identify uh, a low complexity library which could result from too many cycles of PCR amplification or too little starting material. So basically, it will tell you how many duplications are present in your sequences, and your blue line is showing you basically the total number of sequences here, and your red line is showing you duplicated sequences. So on the lower end, you have a certain number of basically reads which have uh, quite a good amount of sequence duplication level. And also, this is basically the reason for this is basically your overrepresented sequence. So there's, for example, take this sequence. For example, this appears in your reads 2500 times, but there is no possible, there is no basically alignment to the no, sorry. Basically, you have too many number of times that this particular read is coming out. So if this is not from a repeat regions, what it means is basically you have a huge duplication in this region, which is normally not the case, because if you have something like that, you will know. I mean, as a person, if you have it, you'll either develop some sort of disease or any type, different type of phenotype. It will not be normal. So intuitively thinking, this is not possible, but you'll also find these kind of sequences which are having very high counts. uh, these are repeating all across your sequencing reads. So what this might mean is basically, it's basically contamination. This can be a vector or adapter sequences. Remember, in Illumina we used adapter. Now the adapter sequences might be similar, so you can have like a lot of adapter sequences which are contaminating your DNA. If the GC percentage was off in the above module, this table can identify the source. Okay, like from what the GC percentage is getting shifted, due to what reason, due to what sequences. If the percent GC was off in the above module, this table can help you identify the source. If not listed as a known adapter or vector, you can just use the sequence and use BLAST to identify from which organism this sequence came from. So you can have an idea how your samples or sequences have been contaminated. Okay, so this was all about FastQC, and after this, after you have done your data quality checks, I understood using FastQC what is your quality of the data, what is your GC percentage of the data, what are the different aspects of your data, basically how good your data is. After that, you need to do some simple pre-processing. So once there are two types of pre-processing here. One is duplicate removal, and basically this is the part that comes in data pre-processing, and another part is data trimming, where you like cut off certain portions of the sequence because they are either low quality or they are adapters or something like that. So uh, that comes under data trimming. So for pre-processing or duplicate removal, you can use there are softwares given. So this is one such. This is basically mark duplicates Picard. Uh, this is a GATK module as well, as I was saying. So if you are interested in, please check this out. This will give you a lot of information about how they do stuff. So, but yeah, the software is Picard and the function used is mark duplicates, and you can use it like mark duplicates, remove duplicates, and it'll remove all the duplicates there. You can use also SAM tools. It is another software that is widely used. And the function is marked up. So the command goes like marked up hyphen r, and then your FASTQ file, it'll remove all the duplicates that is present in your file. So this was one part of pre-processing, which is duplicate removal. The other part is data trimming. Why do you need data trimming? Because you saw these contaminated sequences and all. These might be adapters or anything. So you need to remove these sequences from the data so that you can have a better downstream analysis. So first use case for data trimming is removing adapters. So uh, this step is important for projects with DNA RNA inserts that are shorter than the sequencing read lengths. When performing paired end sequencing, the overlap between forward and backward reads can be used to identify the excess adapter sequences. There are many adapter trimming tools available. The most widely used is one of this, what is called as Trimomatic. It can identify these adapters and this can remove it. There is one module in GATK as well, mark at Illumina adapters as well, where it can mark those adapters, and then after that you can use SAM tools on any other software just to remove those. So there are multiple softwares available to do this stuff. And after removing adapters, you also remove low quality bases as well. So whatever is your cutoff, you keep a cutoff of that and remove all the bases that are below that quality. So your data now is good quality. You're confident about your data, and your data does not have any kind of contamination. Till now, we just have checked uh, from the signal till generating a good sequence file, what are the different steps involved and what are the different kind of softwares you can use to identify and remove certain contamination and other stuff. But till here is basically just getting your sequence file ready. Your sequence you have till here, if you do till data trimming, what you have is basically a FASTQ file with good quality basis, uh, no duplicates, you have removed contamination and everything. So basically what you have is a very good set of data which is like with good quality reads. After this we'll come to the next part in the next week. So after this we'll do variant calling. You'll identify what are the different variants in the genome, what are the single nucleotide variants, what are the structural variants, what are the different type of variants present in the genome. So till this part was identifying the different data formats and like using quality control to basically clean up your data. That is all. So this was the revision for the second week. So now we'll solve some MCQ problems. So you can put your answers in the chat box or like unmute yourself and speak if you want. But I'll put these questions on for a minute, then I'll discuss the answers. Good. One, Okay, let's see. So, the answer is B and D. So I think some of you said A, B, D, and one person I think said like C. I think B and C. Just to be clear, data trimming and data processing and duplicate removal was is different. So you're removing duplicates, that is part of duplicate removal and pre-processing. Trimming is basically just like trimming up your read. So if you have adapter sequences on both end, you're just trimming up that read. You're not deleting the whole read in a whole, you're just trimming that read and making it shorter. So you like when you're trimming it from the ends and removing the adapters, it can be that, or you can just remove individual bases which have low quality scores. So that is the whole purpose of data trimming. And trimming is done on paired end sequencing, because you can guess an idea about the uh, have an idea about the length of the adapter by checking, because in Illumina you'll have paired reads, like one from the beginning, one from the end. You can check how much these are overlapping and what part of the genome they are covering. According to that, you can have an idea about what is the adapter length. So in paired end sequencing, you can like do this data trimming and adapter removing very efficiently. So the answer is basically, yeah, to remove data with poor quality score and for paired end mapping. Is the D1. Sorry, to remove the adapter sequence. Sorry, the D1 is basically to remove the adapter sequence. So, but for paired end mapping, you can do it very nicely, but it is not only limited to paired end mapping. You can do it anywhere. But in paired end mapping, if you check the amount of overlap, you can check how much of it is actually the adapter and how much of it is actually the read. So to remove the data with poor quality score and to remove adapter sequences, data trimming is used. Okay. It will take a minute. Okay, let's look at the answer. It's quite straightforward. So the coverage, if you remember, is given by the formula read count, the average read length by total genome size. So you're basically genome size of you're asked basically genome size of X. So the read count of X is given by this, basically 10 to the power 9. Average read length is given by 150 base pairs, so it's 10 to the power 9 into 150 divided by, you're assuming a 30x coverage. So you're trying to find the total genome size. So if you take it on the left, your coverage will come down here. So this is how your expression looks like: 10 to the power 9 into 150 divided by 30. If you solve this, it comes as 5 into 10 to the power 9, or 0.5 into 10 to the power 10. So you're asked in this form, a into 10 to the power 10. So your answer would be zero point 0.5. Right? Any issues with this problem? If you face any issues, please unmute and ask. Okay, I'm assuming you have understood. So moving on to the next one. In the similar way, find the genome size of Y. Okay, let's see. Do we have any answers? I'll give you another minute. Okay, let's see. You missed this part of the question. The first sample was sequenced with single end sequencing, and the second sample was sequenced with paired end sequencing. So basically, your read length here will be 150 into 2 compared to just 150 here, right? So it's 10 to the power 11 bases, 150 into 2 base pairs. So basically 300 here, and divided by your coverage, 30. It gives you 100 into 10 to the power 10, right? I think you missed the 2 here. Okay, yeah. So keep this in mind. So if it's a paired in sequencing, if your one of the read length is 150, then for the total read length will be 300, because you're sequencing from both ends, right? Okay, moving on. Okay. So what is given here? Your it is given that your genome size of Y was 10 times that of X, and if there are 10 to the power 11 reads generated with a read length of 150 base pairs, the coverage is given by Cx. So what is the value of C? Your basically 10 to the power 11 reads is given here, and instead of it's basically read length of 150 and with paired end, so it's into 2, and you're comparing it against basically the genome size of X, so it's 10 into zero point 0.5 into 10 to the power 10. So this 0.5 into 10 to the power 10. So 10 into zero point 0.5 into 10 to the power 10. So if you solve this, you get C as 600. So it's given that the value, it's given instead of giving you the whole genome size, they're giving you that it is 10 times that of X. So you just do a 10 into zero point 0.5 into 10 to the power 10. And you can easily find the coverage. Okay, moving on. Do we have any answers? The correct answer is B. As we showed earlier, trace will show many peaks, but also a lot of other peaks that run along with the back bottom, which can interfere with the base quality, and the quality scores of this peak are pretty low. So basically, your peaks will not be very distinct. It will be flattened out because of the noise that's coming from other sources. So, so
Uh,
basically, it will be like something like this. Somebody was asking.
yeah, there is one doubt, if you
Yeah,
don't mind.
go ahead.
The genome size, which the previous question,
Hmm.
we got the answer of 100 and of the second question and 600 for the continuation of the question.
Yeah.
Yeah. So we have been shared a PPT, whoever have registered
Hmm.
uh, on this assignment.
Okay.
There it was given that the B is 100 and C is 600. Coverage is 600
Huh.
and sorry, the coverage is 300 it was given. Okay,
Oh.
and the B was 50.
Okay. But according these to these values, it the answers look right. I'm not sure if the values are same, exactly same in your PPT as well.
It's
Kindly
this.
check that.
Yeah, it's the same. It's the same actually.
Okay. But
Can you just cross verify this and
okay.
later let us know?
Okay. I will because I do not have access to the one that has been given to you, so I have to verify it anyway. Okay. I'll verify and let you know. But according to the values here, this look fine to me at least. But
Yeah,
okay.
initially this was the doubt in the previous session, which we attended this Saturday. These three
Okay.
questions were very doubtful. So we came up with three answers. Then later in this PPT, it was uploaded that it is 50. 50
Okay.
into 10
Okay.
to the
Okay.
power 11 or something like that.
Okay. I'll take a look. I'll take a look.
Yeah. And the coverage was 300 because the girl who took our session, she actually uploaded the assignment stating that this was wrong, highlighted it as this was wrong, and this is the right answer. And uh, so,
Okay. I'll check. I'll check. I'm like these assignments have been given to us. I'm discussing from that perspective. So I'm like this might be wrong, but it looks okay to me. I'll cross check and I'll verify and let you know in the
I
next.
just want to know what is the right answer because I'm also confused.
Okay, just let me do it. Just give me a minute. And
Yeah, sure.
these two, right?
Yeah, the first is point 0.5. That is correct. The first
Okay.
question number three. Then it's not 100. It's shown as it's 5 into 10 to the power 11 or 15 into 10 to the power 10. Then the coverage was 300x.
Okay. Okay. Let me check. I think might be some IA issue. Okay. Yes. I have to cross check because this looks right to me at least. The values. I'll cross check and let you know because
Yeah, okay.
according to this, it looks right to me that currently at least. I'll let you know. I'll let you know. Um, apologize for this one.
Yeah,
I
yeah.
wasn't aware. Okay. Okay. Moving on. This might take some time. We're coming to the end of the session, so I'll just quickly go through this. So it's quite simple. So if you have an accuracy, say 95, 98, 95, 97, whatever it is given here, what you do is you find the error percentage. So it's just 100 minus this term. So it's 2, 5, 3, and if you convert it to decimal instead of percentage, it's 0.02, 0.05, and everything. Now you have the percent like probability of error. If you recall, the quality score is given by minus 10 log 10 of that error probability. So you just take a negative log 10 and multiplied it by 10 with the error probability. So it comes, these are the values that comes to, like you can calculate them. And if you round it off, it becomes like 16.98 becomes 17, 13.01, 13, and so on. This is your rounded off quality score. Now for the Fred score, Fred is generally in a plus 33 encoding format. So just add 33 to that. So if you add 33 to all these values, it comes as like 50, 46, and dot 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 whatever the values are coming as. Now you want to convert this Fred values to the ASCII characters. So if you go back to the table, this was the table 50, 50 is somewhere here, so 50 is 2. So your ASCII will be 2. 46 is here, 46 is given by a dot, your ASCII will be dot, and so on. So it's quite straightforward. If you're given the accuracy, you find the error, and then you convert the error to the quality score using the formula minus 10 log 10 of that probability of error. You have the quality score, then add 33 to that to get the Fred score, and then you convert it to the ASCII. So the workflow is quite simple. The conversion of Fred to ASCII, you, it's not possible to remember all these characters. So obviously, you'll be given a table like this, and you can just look up these values and what the characters correspond to. And just put it there. So this will be the uh, and if you can like average this out, it's basically your average Fed quality score. But basically, you want to uh,
I'm sorry, is it the average of Fred plus 33 or just the Fred score? Just
no,
the
it's
quality score.
just the Fred score. It's not the Fred plus 33.
Yeah, that's
This one,
for our
this one,
understanding. Okay. Yeah.
this one. Fred plus 33 is the encoding format, but you're
Yeah.
basically this is your rounded off Q value. This is what they have averaged out, not this one. This is also, I think, is a typo issue. This question also has an issue,
Okay.
but this is the average of this one, not this one. Yeah. So basically, the idea is to the quality scores. This is the important part. Remember how to do that. Accuracy to error to your quality score. Right. Okay. Oh, and yeah, by the way, last time some people asked me to share the link of the places where basically the PPT is. So I think I can share it with you now. So I'll just share the folder drive. So this drive has the last week's PPT in it. I'll upload this week's as well. So you can use it from. Okay. Yeah, it's basically higher the Fred score, lower is the probability of base calling. I'll just quickly go over that table just to make you remember. Yeah, sorry. This is what it means. It's Fred score of 10 means you have a 10, error percent of 10, accuracy 90. The higher the Fred score, the lower is the error rate, the better is your accuracy. Okay. It's actually both. Yeah, the third option is correct. But when contamination happens in the sample, what you'll find is more like the same DNA getting repeated and repeated because like after the contamination has happened, you might have done PCR on that, so that amplifies that contamination. So if you, in your sample you'll see that particular read coming up a lot more, but it's not actually part of your genome. So contamination can also lead to overrepresented sequences, as in the first QC as we saw. So both are the reason. Contamination can also lead to overrepresentation and also diversity. If it is less than normal, then obviously it is intuitive that it will be overrepresented certain sequences. Okay. See. Okay. Correct answer is D actually. So if there is more GC content, it's nothing to do with like trimming of the adapter, because at no point of time like there is a thing to say okay, like the adapters will have more GC. So it's not trimming the adapter won't help you basically lower down the GC content. It's not the source of the GC deviation. There might be overrepresentation. This might be a correct answer for this, but there also like the overrepresented sequence must have high GC content at least. So, and like the amount of overrepresentation should be high, and what it in turn can mean if there is contamination. So the ideal answer is D, but you can also have an argument for C as well, because due to the contamination, there will be overrepresented sequences in the data. So ideally, like it should be both, but the actual reason is basically contamination. Okay. Okay. I'm hoping that's for the above question. Yeah, it's bad quality, as you can see. So, and in the initially, it started off at least decent if you had a median around like 24, 25. But as it went on, you see the box plots became like more broader in number. So basically, what the box plots indicate are basically this line is your 25th percentile, this line is your 75th percentile, and the red lines are your 50th percentile. So your median basically red lines. So as you can see, the quality scores are dropping as you go on the positions. Initially it was okay. There are some sequences which have certain highest quality scores in these regions, in these positions, but maximum of your reads are like you can your median is coming below 20 for maximum of your reads. So it's not a good quality data. You should not you should resequence or do something else with it. Yeah. It's quite simple. You have to check at the position how many reads are covering that position. So for position 1, it's 1, 2, 3, 4. Position 2, it's 1, 2, 3, 4. For 3, it's 1, 2, 3. And for 4, it's 2. So basically, you take the total and divide it by 4. It gives you 3.25. So yeah. Okay. So it's basically you have to put in the formula. So if you are calculating the error of the base, it's just basically 10 to the power uh, log of that, basically the Fred score by 10, and minus will be because initially when doing the Fred score you're taking a negative. So this is the form in which, so if you calculate the probability of error of base 1 using value 30, it comes at 0.001. If you calculate it using 35, it comes as this value, 3.16 10 to the power minus 4. You take an average of this, and it comes as 6.5 of 10 to 10 to the power minus 4, which is basically the value of d, which is 6.5. It's quite straightforward. Okay. Okay. Are the both are both the bases good quality? Check the values again. 30 and 35. Are they good quality? The answer is yes, they are. Okay. Because 30 and 35 are actually very good qualities. Yeah. This is quite straightforward. You are given the size of the genome, you're given the number of reads, you're given the average read length. So your number of reads into average read length divided by the total size of the genome will give you the coverage, depth of coverage. It's basically 2.5 million into 100 divided by 5 million. So it comes out to be 50. So 2.5 into 100 divided by 5. It's out to be 50. Okay. It's also quite straightforward. If you have any questions, no. Okay. This also you have to just put in the formula, and I'll not like take much time using this. So it's basically you're given the accuracy. So if you want to calculate the Fred score, you need to find the quality. So you need to find the error. So the Fred score is given by the error. Quality score is given by the error. So you have an accuracy of 0.9. So your error at probability will be 0.1, and your Q is basically your Q is basically 10. If you just put in the formula of like minus 10 log P is basically 0.1, and if you take the anti-log, you get Q equal to 10, and you add 33 to that. It's basically 43. These are quite straightforward. You just need to put the values in the formula and you'll get the answer. Okay. Yeah, this is the last question. D is correct, but A is also correct. For paired end sequencing, the coverage is counted twice for the mate pairs. If you remember this one, this sum, the answer was disputable, but if you check, we calculated it for twice. So 150 into 2 because the second sample was uh, paired end. So for the paired end sequencing, we calculate the read length as twice as the initial uh, read length. So it's actually both A and D. Okay, guys, that's it for today. We have exceeded our time by quite some time, 15-20 minutes. Sorry for that, but yeah, that's all for today. If you have any questions, you can ask, or we'll end this call and we'll meet next Tuesday. And this video will be uploaded on YouTube, so you can just go and check it out, and you can search by my name. I've put my name here somewhere. Yeah, put it again. You can go to the and search for this name on YouTube, and my channel will come up, and you can find all these videos here. Okay, and I have also put the link to the drive as well for the PPT. You can check it out. I'll upload the PPT after this. Okay, guys, then thank you for joining, and meet you next Tuesday 5 p.m. week 3. Okay, thank you, guys, for ending the call.
Thank you, sir.
Yeah, thank you.